Our next speaker finished PhD in statistics at University of Maryland, Baltimore County, where she worked on multiple testing controlling for false discovery rate with applications to genomic data. A paper she co-authored entitled Oncodomains, a protein domain-centric framework for analyzing rare variants in tumor samples, won PLOS Computational Biology Research Prize in 2018 for its public impact. Prior to that, Dr. Gauran already received a third prize in the 2013 Jan Tinbergen Prize for Outstanding Young Statisticians from the International Statistical Institute. She finished BS and MS Statistics degree at the School of Statistics, proud advisor here, where she is now Associate Professor and Director for Research. Please welcome Dr. Iris Ivy Gauran, who will talk about local false discovery rate for sparse count data in the discovery of hotspots in protein domain. Dr. Gauran.
Good morning, everyone, and good evening to some. So I am Iris Ivy Gauran from the UP School of Statistics, and I will be presenting our work entitled Bayesian Local False Discovery Rate for Sparse Count Data with Application to the Discovery of Hotspots in Protein. This is a collaborative work with my PhD advisors at UMBC, Dr. Junyoung Park and Dr. Dohan Park, with our colleague Marisol Khan from the Department of Computational Biology and her students Ilya Ratsev and Tom Peterson. So in this talk, I will try my best to tie in together three related and that we did as it relates to hotspots. So the motivation of our study is in protein domain analysis as it is applied in cancer. So as we know, our fight against cancer is hindered by its very heterogeneous nature. Cancer arises when there are genetic mutations in the cell, which leads to abnormal growth and eventually to a tumor. However, the dilemma is that traditional methods for identifying somatic variants that drive cancer are gene-centric in that they only consider somatic variants within a particular gene and make no comparison to other similar genes in the same family that may play a similar role in cancer. So in our study, our initial work aims to coin and propose the term oncodomain hotspots, which is a new domain-centric method for identifying clusters of somatic mutations across entire gene families using protein domain models. So we know from the central dogma of molecular biology that DNA makes RNA and RNA makes proteins, and that the information is passed through that pathway. But that's no longer the case now in the presence of the field of epigenetics. So in our study, we were able to look into the overlap of the oncodomain hotspots with the active sites of a catalytic domain of protein kinase, also known as CD00180. So in figure A, we will see the active sites or the sites which are frequently mutated among different types of cancer. And in figure B, we will see the literature-based active site residues. And overall, in our results, we're able to show that there's an overlap between this. Another application that we have to protein domain data is by analyzing at least 5,800 patients from the Cancer Genome Atlas and the following patterns of protein domain hotspots on the calcium binding domain of epidermal growth factors, or CD00054, was observed. So as we can see in green, those are three residues that are known to bind to calcium. And in our study, we were also able to identify them as hotspots. In addition, we were able to identify 42 other residues, which are in blue, which did not bind to calcium, but tend to cluster around the binding pocket of calcium. And in this case, these positions were highly mutated for many patients. And in particular, one of our colleagues, Tom Peterson, discovered that known cancer variants tended to cluster at specific positions more than variants involved with ag-related diseases. So the main question now is, why did I start with the end in mind? So I started with a result or a set of results that we have for some of our work. And the question really is, in the process of understanding cancer, we wanted to look into the level of protein domains. So that further leads to the question that if we're not very familiar with the topic, so what are protein domains? So protein domains are unit of protein structure which evolve, function, and exist independently of the rest of the protein chain. As I mentioned in the previous slides, an example would be catalytic domains, or CD00180 and CD0054, and both of which are catalytic domains of protein kinases. And we're interested in them because they have been implicated in the development of various diseases, including different types of cancer. So let me shift gears a little bit and take on my statistician's hat. So let's look at what the data would look like in our perspective. So consider a given protein domain, and in a specific domain, there are big N positions. So the data will look like a vector A sub n, where the ith value is the number of mutations in the ith position. So this is what CD00 will look like in terms of its bar plot. So we can look at the bar plot because it's interesting to highlight certain features of the data set. So the first feature that we can see is that there are a lot of zeros. So that would mean that there are a lot of positions in the domain with 0 mutations, which is kind of good for the patients involved. Another thing is, it is not surprising to see a lot of ties because we're dealing with count data. So we can see several positions with 0 mutation count, several positions with 1 mutation count, and so on. Another thing that we can observe is that there is a possible overdispersion as it relates to Dr. Peña's discussion earlier. There is a possibility of overdispersion that this data might display. And lastly, in terms of providing a direct-to-the-point assumption on the model that is probably involved in this study, it is a zero-inflated discrete distribution. So the main overarching objective of the study is to test which among the positions are background mutations, which among the mutation within the positions are background mutations, and which among these are related to the disease, or what we refer to as functional disease mutations, while controlling a given level of type 1 error, such as the false discovery rate. So the main dilemma in testing this set of hypotheses is, how do we specify what F0 is? So we know AI is the number of mutations in the ith position, but what does the null distribution look like? So to be able to proceed with the testing procedure, we need to make certain assumptions on the null distribution F sub 0. So here, we were using the idea of empirical null estimation. That is, F0 is estimated based on the data rather than some predetermined distribution, such as the standard normal distribution. In addition, we will use Efron's zero assumption, wherein most of observations concentrated around the center are considered to be null observations, but those towards the tails will be considered or will tend to be non-null observations. So the main dilemma for us is to extend this in the discrete case. So in our case, our main assumption is that the mutation count, which belongs to the set 0 to capital C inclusive, for some unknown integer C, is generated from the null distribution. So to visualize this, our one-stage procedure proceeds by specifying the mutation counts from 0 to capital K, where capital K is the maximum number of mutations in a given protein domain. And then our goal is to estimate C by C hat using the EM algorithm for truncated data by McLachlan and Jones. So once we are able to identify C hat, every mutation count from 0 to C hat is considered to be background mutation, and we do not reject the null hypothesis Otherwise, if the mutation count is from C+1 to capital K, we will test using the local FTR. So an extension of this procedure that we proposed is the two-stage procedure, wherein after identifying the value of C hat using the estimation procedure, by the way, all the estimation procedures are available in our paper, and I will gladly talk to anyone who will have questions about it. In the interest of time, I decided to just summarize the proposed methodology in this presentation. So after identifying C hat, our goal now is to identify D hat. So analogous again to the work that Dr. Peña is discussing earlier, we are looking into the tail probabilities of the zero-inflated generalized Poisson distribution, which is our null distribution for the mutation counts. And we know that from literature that this generalized Poisson distribution with the zero inflation parameter accounts for the overdispersion in the count data. So the tail probabilities used in calculating D is actually based on the large sample theory. So it requires the sample size or the number of positions in a domain big N to be large. Now, what we do is similarly, we do not reject the null hypothesis if it's a number between 0 to C hat, and we automatically reject if it's from D hat to K. For values between C+1 to D hat, we test it using. So with this study, the question is, why did we decide to go Bayesian? So why go the Bayesian route apart from the cliche that you do the frequentist route and then eventually you do the Bayesian route? We really have a very valid and solid reason for proceeding this route because there are two types of protein domains. So there are domains which have large number of positions ranging from the hundreds to thousands, and there are positions with very few there are domains with very few positions, which are even lower than 100. So an example of this is the Helix-loop-helix domain, which is a catalytic domain again. HLH domain is very helpful for the processes of neurogenesis, sex determination, and gut development. So understanding the behavior of this domain, no matter how difficult it is to study, is very important for us. So if we look at the plot, it kind of looks the same as the previous bar plots that I've shown earlier. The main difference being the number of counts here is, the total number of positions in this domain is 93 only. And as we can see, it's still zero inflated, but it displays not only a small number of positions, but sparsity as well. So we wanted to use the smoothing property of Bayesian approaches in order to address this problem. As a response, if we just use the large sample approximation assumptions that will not hold for sure, C is underestimated in all of our numerical scenarios prior to running the full Bayesian scenarios, and pi0, which is the proportion of the null observations used for the calculation of the local false discovery rate, is underestimated. In essence, this will consequently lead to inflated false discovery rate. So with that said, we proceeded with some histograms. So as you can see here, this is the simulated version of the data that I have shown here. And we tried to simulate it using the null distribution, which is ZIGP, and shifted geometric as our alternative or non-null distribution. So we are showing three cases for this one. So the first one, when C equals 5, all gray values represent true null observations or true null mutation counts, whereas the black values represent non-null counts. So that means all black values should be rejected for us to have a good power. And gray values should not be rejected so that we won't commit false positive error. So in this case, the significant values will just start from 6 up to the maximum possible. Whereas in the second case, we start with 2 onwards. So we just reduce the possible values of C. Instead of it from 6 onwards, we allow smaller values of C to be even considered as significant functional disease mutations. And the most important test that we did for the series of numerical studies that we employed in this paper is that what if we are testing the key assumption on the existence of C? And what if there is no C in reality? So that means even some of the positions with 0 mutation count can be attributed as functional disease mutation. So that kind of does not make sense for me without that much biology background, but we are just testing it to check the robustness of our assumptions. So here is the table of a sample of results, some results that we have comparing our proposed non-parametric method versus the existing empirical based method that we did when we assumed that the number of observations is large. So as we can see here, the true value of C is 5, and the procedures that we are comparing assumes Dirichlet prior, and both of them are near continuous uniform priors. And then we compare that with our 2-stage, 1-stage procedure, a very popular stories procedure in the multiple testing realm as well. So we assume a 0 inflated model for our null distribution. It could either be ZIGP or the specific case when we don't account for overdispersion, and that's the 0 inflated Poisson distribution. R represents the number of rejected positions, and FDR represents the false discovery rate, while true positive rate corresponds to the empirical power. So in this case, we can see that for all values, under the assumption of ZIP without accounting for overdispersion, will yield to inflated false discovery rates. Whereas when we assume that there is overdispersion in the model and we account and estimate for that, then the value of the FDR is controlled given that our true nominal value is 0.05. So this is the actual result with the Helix-loop-helix domain that I have shown earlier. So this is for the real data result. As we can see, the empirical based method yielded 7 rejections. Our proposed non-parametric method, together with other methods such as semi-parametric and parametric Poisson and parametric generalized Poisson, yielded NP yielded 1 rejection. And we can see the orange vertical line to be the cut-off point for the EB model. This is under the assumption that everything after this cut-off point are rejected. So I'm not 100% sure it's very clear, but we can see 3 mutation counts here, 2, 1, and 1, and that will add up to 7. Whereas for the non-parametric method, only 1 value is rejected. So that is true for HLH. And then this is for sushi repeats, wherein in this case, we can see an even clearer distinction among all the methods. So in this case, we can see that the orange, which is the earliest cutoff, corresponds to the empirical base. Then the red one, which is the next cutoff, is for the non-parametric method. And then the most conservative in this case is our semi-parametric method. For curiosity purposes, the parametric and non-parametric assumptions vary in terms on how the overall mixture distribution F, or the alternative distribution F1, is specified in terms of their priors. And another application is another epidermal growth factor domain. And in this case, we can see that there is the value for empirical base and non-parametric result method coincides. That is true regardless of the null distribution. So there are some cases that the non-parametric method in a way converges to the result of the empirical base. So all of this are part of our body of work. And the most recent result that we were able to look into is in another catalytic domain, CD00083. And in this data set, we were able to find position 15 of the HLH domain, which is highlighted in red. And this was identified by both methods, empirical-based and non-parametric method, as a hotspot, whereas the other blue parts are identified by the empirical-based method as hotspot as well. So the red part is actually a proven DNA binding position, which verifies the results that we have in this study. So looking into everything that we've done in this body of work relating to proteins and coming back to the Philippines after my PhD graduation, I am now working in terms of proteins as it relates to lung cancer in coordination with the Lung Center of the Philippines. And we're also studying it in the protein level. So the applications of this one, while slightly, the schedule is moved slightly because of the COVID pandemic, is still being applied to the Philippine scenario and in a specific cancer, and that is lung cancer, as funded by DOST. So with that said, these are my references. And I would like to thank Ma'am Gisela and Sir Erniel for organizing this talk. And special shout out to Sir Mario Santo Domingo and Ma'am Leia Tolosa for always inviting me into the PAASE events, especially when there's tons of food during the time that I'm still a graduate student at UMBC. Thank you po.
Okay, thank you very much, Iris. Please reserve your questions for later, or better yet, you can type it at the Q&A box. We will read it later on. So let's